Hi everyone, I'm Dr. Preeti Sharma and welcome to the YouTube sessions for the Microbiology Crash Course. This is particularly for the FMG students who have their exam coming up. However, all the NEET PG and the next aspirants can definitely benefit with this rapid review of the course that we are going to do. So we will be covering in a sequential order all the different chapters and important topics of microbiome, starting from general, going on to fungus, bacteriology, virology and parasitology. Alongside, we'll be practicing all the questions that are important for the FMG students, especially in terms of their PYQs. We'll be also talking about a lot of images, how to identify those images in the exams, and alongside we'll be completing our theory. So all in all, the most high yielding topics and important points from your previous year will be discussed. So what do we start off? First and foremost, we need to know about the historical aspects. Especially in the FMG exam in the past years, there have been questions from scientists. From the history point of view, there have been a couple of questions, and that is exactly what we are going to cater to in the first part of the session. So let's begin. The number one scientist, who's one of the most important that we have, is Sir Louis Pasteur. Well, you also have an image in front of you for Sir Louis Pasteur, and he's definitely contributed immensely to the subject. So what all has he told us about? You can see from the surname itself. When we talk about Pasteur, obviously what has he given us? He's given us pasteurization of milk, which we'll be talking about again in general microbiology. There's also an institute set up on his name that is referred to as the Pasteur Institute in Paris, and that is all that you have from his name, that is Pasteur out here. Whereas what do you have from his first name, that is Louis, L for Louis and L for liquid. Sir also gave us about the liquid medium. Now we are going to read a lot of media today, and you will actually realize that all the media and culture media that I talk about are solid culture medias, like the blood agar, the chocolate agar. They are all solid culture media. But Sir Louis Pasteur in the primitive earlier days gave us the first liquid media with which further advances were made. So what all have we learnt up till now? We know about Sir Louis Pasteur giving us pasteurization of milk, naming a Pasteur Institute of Paris, and L for L, Louis Pasteur for liquid media. What else? Guys, he's also known as the father of microbiology. He's referred to as the father. And when we say father, we can take every alphabet out of father and come to a particular answer. So when we say father, the F will tell us about the fermentation principle. He's given us the principle of fermentation. When we talk about AT, he's given us AT autoclave. Autoclave was described by Sir Louis Pasteur. And when we talk about THER, he's given us one theory, and THER for theory. And which theory has he given to us? He's given us the germ cell theory, or the germ theory has been given to us, and he said that germ theory is something that you have to accept, and abiogenesis theory is something that you have to reject. So one theory for acceptance, one theory for rejection. Germ theory for acceptance and abiogenesis theory for rejection. What else? So let's repeat, because we called him father: F for fermentation, AT for autoclave, THER for theories, and finally Sir has given us the CAR vaccines. What are the CAR vaccines? C for cholera, number one, A for anthrax, and R for rabies. Repeating, cholera, anthrax and rabies, their vaccines, not the organisms. I'm repeating, the organisms were not identified by him, but the vaccines for these diseases were definitely started off. So these are all the different kind of contributions that Sir Louis Pasteur has, and we can go on to the next one. So we'll finish all the scientists and then we'll take up the MCQs, right? So the second one that we have, that is Robert Koch. So Robert Koch is said to be the father of modern microbiology. Repeating, Sir Louis Pasteur was father of microbiology. Sir Robert Koch is the father of modern microbiology. And look at Sir's name, surname, because a lot of things are named after it. So when we talk about K for Koch, he's given us something called the Koch postulates. He's also identified the Koch bacillus. What is the Koch bacillus? The Koch bacillus is nothing but TB, tuberculosis, tubercle bacillus. Then when we go to the next alphabet, that is C, he's given us CCC. Three things with C is what he's given us. Number one, he's given us the Cholera Bacilli, the organism cholera, Vibrio cholera has been given by him, which you all have to learn that Cholera vaccine, again repeating, cholera vaccine was given by Sir Louis Pasteur, the CAR vaccines, but cholera organism has been given by Sir Robert Koch. What else with C? Finally, he gave us the solid culture media. Again, recap. We have Louis Pasteur for liquid culture media, Louis Pasteur for liquid culture media, and Sir Robert Koch for solid culture media. What else with C? He's also given us a color or a dye, and that is known as the aniline dye. The color or the dye is referred to as the aniline dye. Coming to the last alphabet, H. H for hanging drop motility. Hanging drop motility testing was also given by Sir. Repeating, first we'll take the Koch alphabet, Koch postulates, Koch bacilli, then we'll take the C, three things with C, that is cholera, culture media, color, and one thing with H, that is the hanging drop motility. Let's move on to the other scientists. Let's come to Sir Paul Ehrlich. Sir Paul Ehrlich, after his surname, there's an organism that has been identified that is known as the Ehrlichia organism. The Ehrlichia organism is named after him. Other than that, please remember, if you look at the last two alphabets, CH tells you he is the father of chemotherapy. So firstly, Ehrlich will tell you Ehrlichia organism. CH will tell you it's the father of chemotherapy. And PA, what is PA? P for prize, A for acid fast. P for prize. What prize? He's actually a Nobel Prize winner. He's a Nobel Prize winner, and what Nobel Prize has he won? For telling us that toxins and antitoxins work against each other. How he standardized that a toxin can be counteracted upon by an antitoxin. So he's standardized the toxin antitoxin concentration, for which he also received a Nobel Prize. So P for prize, and what is A? A for acid fast stain. Nowadays you know acid fast stain with some other name. Nowadays you know acid fast stain as the Zn stain, guys, right? So that is what you have to know, that originally the acid fast stain was given by Paul Ehrlich. Later on, it was modified by Z and N, Zeal and Nelson. Zeal and Nelson modified it, and today we know it as the Zn stain. But originally, who gave us the acid fast stain? It was Sir Paul Ehrlich. Moving on to the next scientist, it is Joseph Lister, and if you read the name carefully, Joseph Lister is how we call it, because he's the father of antiseptic surgery. He's the first one who told us that during surgery you must use antiseptic, or you must use asepsis in the operation theatre. So he's the father of antiseptic surgery. Joseph Antisep is how we learn it. Coming to the fifth one, which you've been reading ever since you were preparing for your undergrad NEET UG days, Anton von Leeuwenhoek. Anton von Leeuwenhoek is said to be the father of microscopy. And which microscopy are we talking about over here? The usual light microscope. So if light microscope was by Leeuwenhoek, L for L, if light microscope was by Leeuwenhoek, electron microscope is by whom? Electron microscope is by Ernst Ruska. Let's do a recap. When we are talking about light microscope, L for L, it is by Leeuwenhoek. And when we are talking about electron microscope, it is E for E, it was given by Ernst Ruska. Then there's another E, Edward Jenner. This also you read, I remember we used to read it in our undergrad days when we were preparing for NEET UG. Edward Jenner. Edward, you'll say, ma'am, this is also E. How will I not confuse it with Ernst Ruska and electron microscope? Edward Jenner was awarded because he gave us the vaccine. The first vaccine, I hope you all remember, the first vaccine was for smallpox. So Edward gave us the vaccine for smallpox, the first ever vaccine that we had. Coming to the last scientist, three names, or three, first name, middle name, last name. Carrie B. Mullis has given us something which has three alphabets again. So the scientist with the three alphabets has given us a technique with the three alphabets, and that is PCR, polymerase chain reaction. Well, that is a wrap up of the most important scientists that we have. And why don't we practice a few questions in order to know whether we've been able to recall this well. Okay, cholera vaccine. Repeating again, there are two kind of questions. Cholera vaccine has been discovered by, or cholera bacillus has been discovered. Over here, vaccine. If you all remember, we had something called the CAR vaccines. CAR vaccines means cholera vaccines. Number 2, anthrax vaccine, and number 3, rabies vaccine, and all these rabies vaccine, anthrax, cholera, they were given by Sir Louis Pasteur. On the other hand, if I would have asked you, cholera bacillus, that is the organism, has been given by what? By whom? Then cholera bacillus has been given by Sir Robert Koch. Coming to question number 2, who is the father of antiseptic surgery? The father of antiseptic, how did we learn it? Antisep, Joseph. So the father of antiseptic surgery is Joseph Lister. Coming to question number 3. Electron microscopy was discovered by. There was a mnemonic for this also. And as you're watching the video, my request to you all, whenever you get a question, try to pause the video, try to give an answer on your own before I start discussing, so that you know what you were thinking and then you know whether you got it right or not. Okay, electron microscope, E for E. So there are two E's over here, Ernst Ruska and Edward Jenner. I hope you remember the answer is Ernst Ruska. Edward Jenner has given us what? Edward Jenner has given us the vaccine, smallpox vaccine. Coming to the next question. Acid-fast stain was discovered by Joseph Lister, Paul Ehrlich, Louis Pasteur or Robert Koch. Acid-fast stain. So if you all remember, correct answer, Paul Ehrlich. Do you remember what all Paul Ehrlich sir had given us? Let's do a recap. Firstly, Ehrlich tells me that Ehrlichia organism was given. Then CH tells me chemotherapy. He is the father of chemotherapy. P tells me he got a prize. What did he get a prize for? Toxin, antitoxin. And A tells me that he's given us the acid-fast stain. So Paul Ehrlich happens to be the final answer. Coming to the next PYQ, guys. PCR was discovered by. Polymerase chain reaction was discovered by 1, 2, 3. Needs to have a scientist with 3 names, 3 alphabets. Carrie B. Mullis gave us polymerase chain reaction. Well, that is a wrap up of all the scientific aspects. I hope all the scientists are clear to everyone and we can move on to the next topic. Now, the next topic is something you probably see in every microbiology and every pathology lab. However, this is probably also something which you never like to switch on because you many a times don't understand what is seen under it. But the examiner is really fond of all the parts that you see in a microscope. So one by one, firstly, what kind of a microscope is it? Guys, this happens to be a light microscope. This is a light microscope and the first question that we'll ask you is, what kind of light are we talking about over here? We have different kinds of light. We have transmitted light, we have reflected light. So what kind of light is being spoken about over here? You'll say, ma'am, look at this. This is the bulb. This is the bulb, this is the light. I will switch on the bulb, I'll connect it to an electrical source. I'll switch on the plug, I'll switch on this bulb and the light will just get transmitted like this, like regular light from any other bulb in the room comes, right? So what kind of a light am I talking about over here? In a light microscope, we are talking about transmitted light. Like any other bulb or tube light or light source, light is getting transmitted. So first and foremost, you understood the first thing and that is known as a light source. Okay, from the light source, the light starts coming. Then what else do we have? You'll say, ma'am, these are all the light rays. Now a bulb will obviously give light in all directions, right? Bulb is going to throw light in every direction, but I want it to be focused. I want it to go towards one area. So what am I going to do? I am going to put out a condenser over here. Can you all see this tiny little structure at the bottom? We label it. This tiny little structure is referred to as a condenser, and there are two things inside it. Remember that tiny little structure which I am putting up, this round black thing, this is a condenser. And do you know what is fitted inside a condenser? Inside a condenser, we have something called iris. Just like you have the iris in the eyes to see, what does iris in the eye do? It regulates how much of light is going in or not. Same way, there is a condenser which has an iris inside it which will make sure how much of light is going through it. Where is the light going? Let's highlight the next thing. The light is going here. What is this square over here? This square is referred to as the stage. We call this the stage of a microscope. Stage is where the hero will be sitting, right? The hero and all the main lead actors will be on the stage. What's the main lead actor? Your glass slide. Whenever you want to see a glass slide, you will be putting it over here. All those glass slides that you get, which you conveniently ignore in your path and microbiome labs, all those glass slides are kept over here. You'll say that, ma'am, everyone will want this at a different level. Stage might be wanted a little up, a little down, because my power of the eyes and your power of the eyes are different, right? So we have these two knobs that they've given to us, the big knob and the small knob. The big knob is said to be the coarse adjustment. The big knob is said to be coarse adjustment. The small knob is said to be fine adjustment. Repeating, the big knob is coarse adjustment and the small knob is the fine adjustment. What else? We have something over here. This is where you are sitting. This is where your eyes will be there. So there's a lens over here which is known as the eyepiece lens. There is an eyepiece lens which is present over here, and this has a magnification of 10 times. It has a magnification of 10 times. Versus, do you see a lot of lenses over here also? That is where the examiner asks you maximum questions. What are these sets? See, over here there was eye, your eye is here, this is where you are sitting and seeing. So this is the eyepiece lens. What are these set of lenses known as? These are said to be objective lenses, and that is exactly what I want to teach you next. If you've learned all the names, I'll teach you the details now. See, let's do a repeat. What you see at the bottom, this is a light source. After that, what you see where the light is going, that's a condenser with an iris. Then the light is going on to the stage. The stage can be moved up and down by the coarse and the fine adjustment. Thereafter, you have a set of lenses over here called objective lens, and then you have a lens over here that is known as the eyepiece lens. Now, what are the other details that you have to know in terms of magnification? So listen to me carefully. You will say, ma'am, one set of lenses that you've told me are the objective lenses, and one set of lenses that you've told me are the eyepiece lenses. And you've told me eyepiece lens is only one and it has a magnification of 10 times. But objective lens is of 4 times. Now that is what requires attention. One lens will only help you scan the slide. Like you scan a page on a printer, like your human eye, a little bit magnified, 4 times magnified, only 4x. That is a scanner lens. Okay, next you have something you'll say, ma'am, I want to increase from scanner. I'll go to low power magnification. Then I'm going to go to a higher power magnification, and finally I'm going to look at the slide with oil. I'll put oil on the slide and I'm going to look at it with oil. So I will then use the oil immersion lens. What is the difference? Scanner was what magnification? 4 times. Low power is 10 times. High power is 40 times, and oil immersion is 100 times. Simple to learn. What is the protocol? 4, 10, 40, 100. Repeating, we have 4, 10, 40 and 100. Scanner, low power, high power, oil. 4, 10, 40 and 100. So what do you think? Now if I ask you, what is the total, or what is the, how do you calculate the total magnification? You will have to add both of these. You will have to add. So for example, you say, ma'am, this is your microscope. You are looking at the microscope from here, your eye. So one is fixed. 10 times will always happen. Then depends which one are you using from here. Are you using the 4x? Are you using the 10x? Are you using the 40x, or you're using the 100x? So for example, I'm using the 4x. So what is the total magnification? 4 into 10, 40. So let's do from here. If I want to calculate total, you'll say if you're using the scanner, then your total becomes 40 times. If you're using low power, then your total becomes 100 times. If you're using high power, then your total becomes 400 times. And if you're using oil immersion, 100 into 10, your total becomes 1000 times. So if I ask you, what is the maximum magnification that you can obtain in a microscope? Think logically. What's the maximum magnification? You will say oil immersion will give me 1000 times, and the eyepiece is going to give me a 10 times. So what is the maximum magnification that we can receive? It is a 1000 times. 100 into 10, 1000. That's one of the most favorite questions that you have in the previous year papers. So that's it about the light microscope. Let's move on to some other microscopes. Let's take up a dark field microscope. So what do you understand by dark field microscope, guys? Please remember, when you're dealing with dark field, you can see the background is very, very dark. Why is the background dark? Because you are dealing with dark as R, so you're dealing with reflected light. Now you know that why I was emphasizing so much on the type of the light, because when you're dealing with a light microscope, light has a transmission to it, transmitted light. When I'm dealing with a dark field microscope, I'm talking about a reflected light. The background will be dark, and anything that is very, very thin, what are you going to use it for? Anything that is very, very thin, like a spirochetes, or for that matter, guys, even flagella of the bacteria is very, very thin. Anything that is very thin can easily be picked up on a dark field microscope. Coming to the next one, everyone I'm sure has seen this microscope earlier. This microscope is the very famous fluorescent microscope. What is the light source that we have for fluorescent microscope? The light source is the UV light. It is the ultraviolet light. So let us analyze a few things. You will say that, ma'am, fine, you've told us that fluorescent microscope is going to do with ultraviolet light, but that ultraviolet light, ultraviolet light you've seen at so many places. Forget this microscope, but ultraviolet light you've seen at so many places, right? Even in day-to-day life, you might get to see in some instrument, in some machine, ultraviolet light is used. But not every ultraviolet light is going to give me this kind of a green color, because when you're dealing with a fluorescent microscope, the light is ultraviolet light, but what you're also using is a shiny color. You're using a fluorescent dye. You'll say, ma'am, which dye are you using? For example, this is a slide. You say that, ma'am, there's a patient in our country. This patient has come up with cough with sputum. So cough with sputum in our country, the first thing that I think logically is it could be a case of tuberculosis. If I'm suspecting tuberculosis, this is the sputum sample that is there on the slide. What is the dye that I will add to it? If you're suspecting TB, auramine and rhodamine are very famous fluorescent dyes. So please remember, auramine and rhodamine are the fluorescent dyes. Coming to next, if you're suspecting fever with chills, malaria, what is the fluorescent dye? Acridine orange is the fluorescent dye. And number three, if you're suspecting a fungus, anyway, fungus has become, once upon a time in the second wave, was quite a hero, second wave of COVID. So fungus, fluorescent stain, remember fungus, fluorescent is calco flower white. Fungus, fluorescent is calco flower white. Repeating, for TB, it is auramine rhodamine. For malaria, it is acridine orange. For fungus, it is flower white. And what will then happen? So when this UV light comes in contact with any of these dyes, the next question that they will ask you in the paper is what wavelength changes to what. So please remember, shorter wavelength changes to longer wavelength. Initially, the ultraviolet light had a shorter wavelength. When it will come in contact with the fluorescent dye, it will change into a longer wavelength. So shorter to longer is what you know for fluorescent microscopy, and that's the final outcome. That's the final outcome that you get. You finally get to see all these fluorescent color organisms, and this was actually a case of TB only. You can see the bacilli. So now if I ask you if this is actually a case of TB, what fluorescent dye must have been used? The fluorescent dye that must have been used must have been either auramine or it would have been. Okay, so this one's also done. Let's move on to the next microscope, the very, very famous Electron Microscope. So first, the picture is quite characteristic. If you look very carefully, if we look at this very carefully, we will see that it looks somewhat like a spaceship. It looks like a space shuttle because it's closed from everywhere. Why is it closed from everywhere? For example, if I keep it here, this is the electron microscope. If I want to peep inside it and ask you what is the source, you'll say, ma'am, for everything you were telling me a light. Let's do a recap. For example, for light microscope, I told you the source was transmitted light. For dark field microscope, I told you the source was reflected light. For fluorescent microscope, I told you the source was UV light. But for electron microscope, there is no light. The source is a beam of electrons. The source is a beam of electrons, and electrons always have to be kept in vacuum. You can't keep electrons out in the open. You have to keep it in a vacuum, and that is why they've created this kind of a vacuum out here. That is why this looks like a closed space shuttle kind of thing. What is the next difference? When you take the electrons and they are in a vacuum, finally you'll say, ma'am, at the end of the day, you have a slide, you have a specimen on top of it. The difference is that in light microscope, when you're given a slide during your practical exams, is that slide made up of glass? Is it a glass slide? Yes, because in light microscope we use light. Light and glass are, compatible. But when I'm dealing with electron microscopes, I can't put electrons on a glass. With electron microscope, I will have to use something called a metal slide, and that metal slide is usually made up of copper. That metal slide is usually made up of copper. So remember, this slide that we are using over here is going to be a metal slide, and finally you will be able to see the picture on an electron microscope. There's one more difference that we have. Actually, that's a very famous pathology question, but even in microbio, they tend to ask you that what is the difference in the fixatives? Like, for example, let's take an example of, say, a kidney biopsy. A surgeon has performed, a nephrologist has performed a kidney biopsy, and all of us know that in kidney we have to do light microscopy also, and in kidney we have to do electron microscopy also, right? Okay, so when both of these are decided anyway, then I would say that fine, this kidney, there are two, from here you've taken one tissue, from this area you've taken another tissue, you have two tissues. I'll say send one for light microscope, send one for electron microscope. So you will put them in what solutions? What containers will you drop first biopsy in? What container will you drop the second biopsy in? What solution? So the one that you're sending for light microscope, you will drop it in a container of 10% neutral buffered formalin. 10% neutral buffered formalin, and in a container which you want to send for electron microscope, you are going to use 2 to 2.5% deuteraldehyde. You are going to use 2 to 2.5% deuteraldehyde. So repeating, for light microscope it is 10% NBF, for electron microscope it is 2 to 2.5% deuteraldehyde. Very, very important questions. Now that we've done the microscope, just a quick one or two questions to assess our knowledge. PYQ. Maximum magnification that you have of a light microscope. Repeating, maximum magnification that you have of a light microscope, 10x, 100x, 40x, 1000x. So they've asked you the maximum magnification, and I think everyone knows, we've just done this, that you have to multiply the maximum of the objective lenses with 10. So 100 into 10, you get 1000x. That's the maximum, right? That solves your question perfectly. Let's move on to the next question. Which of the following fluorescent stains is correctly matched? Which of the following fluorescent stains are correctly matched? Let's go one by one. Acridine orange for TB. Does not sound right. If I remember correctly, acridine orange is for malaria. Acridine orange is for malaria, and TB ke liye we have auramine rhodamine. For TB we have auramine rhodamine. So this is not correctly matched. Auramine and rhodamine for malaria, again a mismatch. Auramine and rhodamine are for TB, and for malaria we have acridine orange. Coming to the third one, calcoflower white for fungus. That is sounding right. Calcoflower white for fungus. Up till now, I think this is a right statement. Coming to the fourth one, acridine orange for fungus, not at all. For fungus, it is calcoflower white. For acridine orange, that is basically used for a case of malaria. This is also incorrect. So what is the final correctly matched statement? The correctly matched statement, calcoflower white for fungus. Okay, these were the two quick questions that we had, and I think now we can move on to the third topic. I hope you guys are not finding microbiology up till now to be a very tough or an extremely lengthy, and it is definitely cramming up, I wouldn't deny it. But yes, a few mnemonics and concepts here and there will help us sail our boat through, right? Okay, let's go on to the next topic of general microbiology, the bread and butter of micro, the different staining techniques. And I think while I've said bread and butter, I'll start with genuinely the bread and butter, that is gram staining. No microbiology lab can be ever made without starting a gram stain. So what do you have? What all questions can you get? Either they'll give you the names of these chemicals and they'll say identify the stain, or they'll give you these colors, pictorial representation, or they'll simply tell you that okay, I've given you these four chemicals, arrange them in the right order. So how do you identify and how do you deal with a gram stain? The mnemonic is come in and stain. If you want to learn it, simple, that's the protocol. If you want to learn gram stain, then you just have to come in and stain. So what is this? Come in and stain. CIAS. That is how we learn it. We call it come in and stain. So what is the first thing that you add? The first thing that you add is crystal violet. You can see a violet color. So please make sure when we add crystal violet, the gram positive organisms and the gram negative organisms, both of them are going to become what in color? Violet, purple in color. So whether it is a gram positive or it is a gram negative, on adding crystal violet, both of them will become purple in color. After crystal violet, I for iodine. Iodine is said to be the mordant. What is a mordant? Mordant is like a hammer. Like if you put a nail and then you hammer it so that the nail goes inside the wall. Same way, if you put a stain, you want it should go to all the bacteria. So you put a hammer on top of it. Iodine is that hammer which will make sure that crystal violet reaches all the bacteria. So that hammer is known as a mordant. Next, you use alcohol. Either you can use alcohol or acetone. Have you seen the color has vanished? Because alcohol or acetone is a decolorizer. Means it's going to pull all the color out. So now telling you, initially the gram positive and the gram negative both had got a purple color. On adding alcohol, gram positive doesn't show any change. Gram positive still remains purple because positive and purple will always go together. The gram negative ones are going to become colorless. What is your mnemonic? Positive people, positivity stays till the end. Once positive, always positive. So remember, positive gram bacteria, the gram positive bacteria which had become purple once, they will still remain purple always till the end. But gram negative, negativity makes you lose everything in life, correct? Exam going students, remember, negativity will make you lose everything in life. So negative bacteria will lose the color, they will become colorless. You say, fine ma'am, now add a color to them also. So there you add the last red color thing that you're putting over here. Safranin. Safranin is the last stain which now you'll say, okay, positive ones from the beginning to the end have remained purple. The negative ones had lost the color in between. So now the negative ones have also become red because what have I got? I'm finally having safranin. Finally having safranin. So this is your final interpretation of a gram stain. So let's repeat. When you look at a gram stain, can you see all these things which are purple in color? All these organisms which are purple in color, they are going to be gram positive. And all these organisms which are reddish in color, reddish pinkish in color, those organisms are definitely going to be gram negative. I hope the entire protocol is clear. Come in and stain. Yes. Coming to the next one. That's another one which you have in terms of the bread and butter of microbiology, and that is the Zn stain. Zn stain, also known as the acid fast stain. Zn stain, also referred to as the acid fast stain. What do you have to know in this, guys? Let's proceed. So there's a mnemonic for this also. So once a student had asked for a mnemonic in class, I was just teaching it generally. The student said, no ma'am, we need a mnemonic. So then we made an impromptu mnemonic, and that was class has asked mnemonic. For Zn stain, that is how we learn it. Class has asked mnemonic. So what is this CHAM? Let's start. What do you add with C? With C, we add something called carbonyl fusion. The first thing that you're adding over here is carbonyl fusion. With H, you can see that there is heating going on. First, you put carbonyl fusion. After that, you're going to put heat. You're going to heat the slide. After that comes A, you put acid. Most of the times you put sulfuric acid. Most of the times. Most commonly added is A for acid, sulfuric acid. And finally, CHAM. In the end, you have a second stain. Can you see something bluish in color? They've added something called methylene blue. They've added something called methylene blue. So repeating, CHAM, carbonyl fusion, heating, acid, and methylene blue. Sometimes, okay, now if you look at the colors, when you added carbonyl fusion, what was the organism becoming? Everything was becoming red. So please remember, carbonyl fusion gives a red color. And when in the end you're adding methylene blue, you'll say, ma'am, the background things are becoming blue. So what are the two colors that you have in acid fast stain? You'll say, ma'am, we primarily have red and blue. And now if we see this particular picture, I think you will be able to guess this very, very well. And what do you have over here? Have a slide, and that slide is classically showing you that the organism, this is a case of TB guys, the organism has come out what in color? So let's ask you, when we ask you in terms of the result, the organism has come out to be red in color, and if they ask you what is the color of the background, you can very well see that the background has come out to be blue in color. So, you know, the examiner is very smart. He won't give you the photo. He'll ask you as a fill in the blanks that in the Zn stain or in the acid fast stain, organism is what in color, background is what in color. So, can we see that the organism is nicely yellow in color, the organism that I've marked over here is nicely red in color, and the background that you're having is blue in color. And this is actually a case of TB. So, the next question coming up in front of you, you used carbon fusion, then you used heat, then you used acid alcohol, then you used methylene blue. Let's write that down once again and see some variations. Repeating, carbon fusion, heat, acid, methylene blue. If I say in the first, particular variation that heating has not been done, I have removed this heating step. You will say, ma'am, if you're not heating, mainly you're doing carbon fusion, then you're doing alcohol, acid alcohol, then you're doing methylene blue. You're not heating. So what kind of Zn stain will you call it? You will now call it the cold Zn stain. You will now call this the cold Zn stain because you're not heating it, right? And this is known as the Quignon or the Gabbett stain. This variation is said to be the Quignon or the Gabbett stain. Repeating, the cold Zn stain is known as the Quignon, KK sound. The cold Zn stain is known as the Quignon or the Gabbett stain. Okay, now having said that, moving on, let's go on to another variation. Firstly, okay, if I ask you, like I think this question must be coming in your mind, that ma'am, why have you removed the heating process? Why have you made it cold? For what purpose? What is the use of the Quignon or the Gabbett stain? Remember, there's a parasitic family, many of you might have heard of, known by the name of coccidian parasites. Coccidian parasites, again a K sound coming. They don't need the heating process. Even without the heat, they can get stained. So remember CC or KK sound. Cold Zn stain is known as Quignon stain and it is used for the coccidian parasites. Let's do one more variation. Again, we'll write it down again. So what do we have? The Zn stain says carbon fusion, then heat, then acid, then methylene blue. Now they ask you what concentrations of acid are you using. So please remember, what are the different concentrations of acid that we are using? Anything where the word TB comes, TB like tuberculosis or any atypical mycobacteria, anywhere the word TB comes, we are talking about 20% H2SO4. We are dealing with 20% H2SO4, T20, right? T20 is how we learn it. So anything to do with TB, we say T20. Next, anything to do with leprosy. What is leprosy? Lepra bacillus. Anything to do with leprosy or lepra bacillus. How many alphabets does lepra have? 5. That means we are going to use 5% H2SO4. 5% H2SO4. Then for things like nocardia and legionella, we use 1% H2SO4. For nocardia and legionella, 1% H2SO4. And finally, if you want to stain the spores of a bacteria, you go in for the least concentration, that is 0.25 to 0.5% H2SO4. So basically I've written it in a decreasing order, right? For TB, you can very well see that for TB, an atypical TB, T20 you will consider. For lepra, you will count the alphabets, 5 alphabets, 5% H2SO4. For nocardia and legionella, it is 1% H2SO4. And for spores, it is 0.25 to 0.5% H2SO4. Last question pertinent to this table is, out of all of these, which of them is known as the phytophoraco stain? Very, very important question. Which of these is known as the phytophoraco stain? So what does phyt stand for? Or what does it sound like? You will say, ma'am, phyt sounds like 5. So which of them was using 5% H2SO4? The lepra bacillus was using 5% H2SO4. So can I say the same Zn stain in which I am using 5% H2SO4, basically I am using it for the lepra, that has been given a separate name called as phytophoraco. So easiest mnemonic, phyt sounds like 5. So the 5% H2SO4 variety is known as the Fight for Aqueous stain for the 5-alphabet Leprobacillus. Okay, so having said that, now the next thing that you would want to ask me that, ma'am, right now you told us TB, leprosy, nocardia, are these the only things that are positive for ZN stain? No, now comes the next thing. What are all the things that you have in microbiology that are ZN positive? Or in other words, you can say that is acid fast positive. So please remember there's a mnemonic for that. And that mnemonic is my nose is cold and hot. How did I make this mnemonic? Because I got to know that ZN stain can happen with heating also. ZN stain can happen without heating also. Did I just tell you that? ZN stain can happen with heating also, the routine wala, and ZN stain can happen without heating also, the cold one, right? So then we made a mnemonic, my nose is cold and hot. So what are the things which are ZN stain positive? My stands for everything with mycobacteria. My stands for everything with anything of mycobacteria, mycobacteria TB, mycobacteria leprosy, or any atypical mycobacteria. As long as the word mycobacteria is coming, you should be happy that yes, mycobacteria for my. Coming to next, no. No for nocardia and legionella we put together. Nocardia and legionella we are going to put together. Then we come to is for isospora, is for isospora, C for cyclospora, C for cyclospora, and one more C, that is cryptosporidium. Isospora, cyclospora, and cryptosporidium. What do they belong to? For those who have not read parasitology, nothing to worry. Let me tell you, Isospora, cyclospora, and cryptosporidium are nothing but members of which family? They are the members of the coccidian parasites. And have I told you that for coccidian parasites, will you use the regular ZN stain or will you be using the cold ZN stain? The Kinyon variety is what you will be using over here. Moving on to the next, my nose is cold and hot. So HO, the HO that you have over here are hooklets of hydated. Please remember HO for hooklets of hydated are acid fast, and T, the T is left. The T is eggs of Taenia saginata. Taenia saginata eggs are also going to show you an acid fast appearance. So repeating, my stands for mycobacteria, nose for nocardia legionella, is for isospora, C for cyclo and cryptosporidium, HO for hooklets of hydated, and T for Taenia saginata egg. These are all the things that are ZN stain positive. One more stain that I wish to discuss at the outset is this one. You will say, ma'am, this is looking a little different because the background has got a color. In everything when I was showing you, I used to say, look, bacteria has a color. In the next photo that I showed you, again for ZN stain, I said, look, the bacteria has a color. But now I'm showing you something different. I'm saying the background has a color. So the background is going to take up the stain. The organism is going to be unstained. The background is going to pick up the stain and the main organism is going to be unstained. So what do you think? Is this a routine one or is this going to be something like a negative stain? So please remember, this staining protocol is a little different. Like you had negatives in a film, negatives in the Polaroid and the other photos that we used to take long back. Now obviously very few cameras are used, but we used to click and we used to get a film, a negative film from which we could develop more pictures. So what is a negative stain? It looks something like a negative film. It looks very black in the background. Negative stain is particularly used for organisms that have a capsule, because you can't put color to the capsule. In many of the organisms you can't put color to the capsule. So what do? You put a color to the background. You will give a color to the background so the capsule will shine out. The capsule will get highlighted that means. Okay. So please remember, this is very commonly used for Cryptococcus. Cryptococcus, which we will be studying in the next session, which comes basically under fungus or mycology. Cryptococcal meningitis we will study. So whenever you want to pick up Cryptococcus, you do negative stain. Any examples of a negative stain that you've heard of? Remember, there's something called an India ink stain. There's India ink stain. There's something called a nigrosin stain. India ink and nigrosin stains are negative stains that we have. Okay, well, the most essential stains that you need for the exam are somewhere done, and I can probably ask you this question now, which has come as a PYQ in the FMG exam. Gram staining is what? Is it a simple stain, a differential stain? Is it a dry heat sterilization, or none of the above? So this much I think I know, that it is not any sterilization technique, it is definitely a stain. So tell me, is it a simple one color stain, or is it helping me differentiate? As far as my thought process goes, gram stain differentiates gram positive organisms versus gram negative organisms, right? It differentiates gram positive versus gram negative, which basically means that this is a kind of a differential stain. This is a kind of a differential stain. Let's move on to the next question. What is the correct order of this gram staining? Think of your mnemonic and then select the answer. I give you 5 to 10 seconds. Select your answer, gram staining. If you remember the mnemonic, come in and stain. Come in and stain. This means the purple color one and the red color one. The purple color one was crystal violet. I was for iodine. A was for alcohol or acetone, and S was for safranin. Come in and stain. That's the protocol that we have for gram staining. Crystal violet will give a purple color, as all of us know, and safranin will give a red color. Coming to the next question. What is the stain used in the preparation of the AFB? Acid-fast bacillus. The stain used in the preparation of acid-fast bacillus. Jimsa, Loeffler's, Ziehl-Neelsen, or India ink. I think we've understood. Acid-fast means we are dealing with the Ziehl-Neelsen stain, none of the others. And right now, one more question that you can get. India ink. India ink is what kind of a stain? India ink is a negative stain, means whenever you want to stain something like a cryptococcus, or you want to stain something that has a capsule, negative staining is what you use. Coming to the next question. In Zn staining, what is the secondary stain? In Zn staining, what is the secondary stain that you have? Crystal violet, safranin, methylene blue, or alcohol? So think of the mnemonic for Zn stain. Class has asked mnemonic. What is a secondary stain? First, let's write down. Class has asked mnemonic means I first add carbonyl fusion. That's the first red color that I add, primary stain. After that, I do heating. After that, I put an acid, which everyone knows, sulfuric acid, H2SO4. And then after that, we add something called methylene blue. So this becomes the next color. This becomes the secondary color. The stain, the color that you add first will be primary. The color that you add second or last will be secondary. Over here, the secondary stain was asked. So the secondary stain definitely becomes methylene. Let's practice another question. Metachromatic stain. Now this is something I'm giving you as homework, because this stain I will be teaching you under the bacteriology section. So in the next section, obviously I will be teaching you bacteriology, and that time I'm going to give you the answer. So this is going to be a little bit of a homework, and if you know the answer to this in this session or in this video only, you can always type out and let me know what are metachromatic granules, which of the following are the stains which are going to stain them, which bacteria am I talking about. So which bacteria and which stain? If you know the answer, put it in the comments. If you don't know the answer, the next session of bacteriology, this is going to be very, very, very important. Okay guys, staining methodology questions are done. I think we can move on to the next one, and the next one is actually very interesting. I'll first make you attempt the question. The next topic is going to take care of this. Mesosomes are formed by the invagination of what? Mesosomes are formed by the invagination of cell membrane. Invagination more prominent in gram-positive bacteria. Both the statements are true. Both the statements are false. This is not a PYQ. This is a new question that we got up, but is something which is definitely expected. So this is about the anatomy of a bacteria. What all does a bacteria have? So what we'll do, first let's discuss the entire anatomy, and then let's come back to the question. The answer to this is both the statements are true. So let's talk about the bacterial anatomy. I've tried to draw one hand-drawn diagram before we go on to something very big. Let's do something basic that we have over here. Okay, so the first thing that you see over here, you'll say, ma'am, let's start from the outermost. This outer pink color thing that you have, that is either a capsule or a slime layer. So obviously common sense tells me capsule will be very very rigid. If the organism has a capsule, it's going to be very very rigid, and slime layer is going to be very very loose. So the organism will have either it will have a tough rigid capsule or it will have a slime layer. So all of you can see that the outside is made up of capsule. Now there's a question on the capsule that you get, guys. Please note, what is a capsule made up of? Remember, capsules are made up of polysaccharides. Capsules are made up of polysaccharides. If I said that, this means there will be some exception. And yes, there is an exception which I'll be teaching you under bacteria, that is Bacillus anthracis or Bacillus anthrax. Remember, Bacillus anthracis does not have a polysaccharide capsule, it has a polypeptide capsule. That's going to be a very important question that we'll be doing under bacteriology. Repeating, all the capsules that you have are polysaccharides, but the one that you have in Bacillus anthracis, that is going to be a polypeptide capsule. Coming back, what do you have after capsule? Look at the inner ones. Look at the black and the green color layers. They are the cell walls. They are both part of cell wall. So this black color one also and the green color one also, they are all cell wall, and that is where 10 star most important questions are going to come. Why? Because cell wall of gram positive and gram negative will vary. I'll show you another picture. Have a look at these photos. I'm sure you've seen them in your textbooks. This is the cell wall of a gram positive and this is the cell wall of a gram negative. So ma'am, how did you identify just within 2 seconds? What made you identify gram positive, gram negative within 2 seconds? Let's see. Let's try to, you remember in sometimes in our free time when we get those things or links on Instagram or probably on Facebook where they tell us to select the differences between two very similar looking photos. So these are two very similar looking photos and I've given them to you to find out the difference. You'll say, now first I'll tell you the similarities. This also has something called NAG and NAM, and this also has something called NAG and NAM. Means N-acetyl glucosamine, N-acetyl glucosamine and N-acetyl muramic acid is over here. N-acetyl glucosamine and N-acetyl muramic acid is over here. So both of them are present in a gram positive and a gram negative cell wall. But then I see something else also. I realize that there is something extra that is present. There is something extra. The yellow color ones are what? The yellow color ones are NAM. So these yellow color ones which are NAM, I am seeing that they are getting connected by 1, 2, 3, 4, 5 molecules. I've also tried to draw it out. See, you've got NAM and NAG, NAM and NAG, NAM and NAG. But in a gram positive organism, you will say that ma'am, NAM is connected. NAM is connected, whereas in a gram negative organism, I don't see any connection anywhere. So what is the point? There is a pentapeptide bridge between the NAM molecules. There is a pentapeptide bridge in a gram positive organism and there is no bridge in a gram negative organism. It's the easiest mnemonic that you can learn. P for P. What do I mean by that? That in a gram positive organism, a pentapeptide bridge is present, and in a gram negative organism, a pentapeptide bridge is not present. Have a look at this photo again. You'll say, yes ma'am, I can see the 5 pentapeptide here, but I don't see the pentapeptide here. So you are right, I can finally spot the difference. Pentapeptide bridge between the NAM molecules, P for P, positive organism, not seen in a negative organism. What other differences do you have? What other differences do we have between gram positive and gram negative? Another P coming up. Peptidoglycan layer. So which of them will have more peptidoglycan? A thicker peptidoglycan. You will say, ma'am, P for positive, P for peptidoglycan. So gram positive organisms have a very very thick peptidoglycan and gram negative organisms have a very very thin peptidoglycan. So you will say that, ma'am, okay, over here in gram positive, majority of it is P for P. It has a lot of peptidoglycan on it. But in gram negative, when the peptidoglycan is very very thin, what other thing makes up for it? What makes the cell wall then? I'll say it is lipopolysaccharides. Lipid. So if I ask you, out of gram positive and gram negative, lipid content is where? more in amount, where you say lipopolysaccharides are more in the gram negative ones. So lipid content will be more in the gram negative ones. Repeating guys, now finally the take home message for you guys is that when you are told to differentiate between a gram positive and a gram negative, remember P for positive, which means P and P is what you are going to write. Which of them has a thicker peptidoglycan? Which of them has a thicker peptidoglycan? It is gram positive. Which of them has the pentapeptide bridge? The pentapeptide bridge is also present in the positive. So positive peptidoglycan and pentapeptide. That's the cell wall, main component, cell wall. Finally, after cell wall, you say, ma'am, this brown color thing is the cell membrane. And can you see? So I'll label it. This brown color thing that you have is the cell membrane. Can you all see the cell membrane has dipped inside over here? And the cell membrane has dipped inside over here. What is this dipping inside? This dipping inside is a mesosome, and that was the question that I had asked you. The cell membrane dips like this and the cell membrane dips like this. So can I say it is formed by the invagination of the cell membrane? Yes. Does it happen both in gram positive and in gram negative? Yes, but it is more in gram positive organisms. Mesosomes are prominent, again P for P. Mesosomes are prominent where? Mesosomes are prominent in the gram positive organisms. But what are mesosomes? होते क्या हैं? What is the purpose of these mesosomes? So please remember everything about mesosomes that you need to know. Remember, mesosomes are also known as chondroids. That's another name that has been given to them. Mesosomes are also known as chondroids. Number one question, they are the cell membrane invagination. They are the invagination of cell membrane. Number two, where are they seen more? They are prominent in positive than in gram negative. And last, what is their function? They function for the respiration. This is a respiratory organ of the bacteria. Respiratory function or respiratory organ of the bacteria is a mesosome. Other than this, there is no fourth question that can come. Cell membrane invagination, prominent in positives, and respiratory function, and hence the answer over here was both the statements are correct. What else am I left with in that diagram? I'm left with these three projections coming out called pili. Remember, pili are also known as fimbria, and their function is attachment. Whenever two bacteria want to attach, whenever there are two bacteria and those two bacteria want to attach, they can attach like this by the pili. So pili or fimbria are basically for attachment. Versus, what is this? Flagella. The next one is flagella. And when we say flagella, these wavy things coming out, I think everyone knows that flagella is used for locomotion. When the organism wants to move around, lot of movement, motility we'll be talking about in bacteriology. When the organism wants to move around, definitely locomotion is something which will be carried out by flagella. Okay, having said that, what is the next question that we have? Here you go. Mesophilic organism grows at what? So mesophilic organism is going to grow at what particular temperature? There are three main temperatures that we have. Want to learn those? We'll come back to the answer after that. Requirements for a bacteria. For a bacteria to grow, please remember, the temperature can either be a medium range temperature or it can be in minus. So they are like psychic temperatures, psychrophiles, psychic temperatures, minus temperatures, or they can be higher temperatures. So when I'm talking about medium range temperatures, 20 to 40, anything between 20 to 40, the medium range, the bacteria that grow in this medium range temperatures are mesophils. Meso will remind you of medium range temperatures. The organisms that grow at minus temperatures, they are psychic temperatures, psychrophiles. Psychrophiles grow at psychic temperatures of minus, and the organisms that grow at high temperatures, 55 to 80, for high temperature we use the word thermo, they are thermophiles. So easy to learn, mesophile medium temperature 20 to 40, psychrophile psychic temperatures minus, and thermo means more temperature, so higher temperature 55 to 80. So the PYQ that came was mesophilic organisms, medium. So it cannot be under the minus, it cannot be low temperatures, it cannot be high temperatures, it has to be somewhere medium between a 20 to 40. So that's the best answer that you have over here. Okay, having said and done with this question, let's move on to the next. The next question that we have, sporulation occurs in this particular phase of the bacterial growth curve. Repeating, which phase of the bacterial growth curve is going to show you sporulation? So I hope some of you definitely know the answer. For those who don't, you will know the answer in exactly 5 minutes, because what we are going to discuss is the bacterial growth curve. So what I'll do, I'll first draw the bacterial growth curve. What exactly are you trying to analyze in a bacterial growth curve? It has a flat line, then the line goes up, then again a flat line, and then the line goes down. So basically, when you're talking about the bacterial growth curve, this axis over here is going to basically tell you the number of the bacteria. This is going to tell you the number of the bacteria. This obviously will tell you the time and the duration. So first you will say that, ma'am, there's a flat line, means this is said to be a lag. This is said to be the lag phase. After that you will say rise in the number. So rise in the number means we are in a log phase. Log always means exponential, increase in number. Bacteria are multiplying. Then comes this, you'll say, ma'am, we've reached a plateau. This is a stationary phase. This line over here is the stationary phase, and finally dropping down is what phase? It is referred to as the decline phase. This is referred to as the decline phase. So repeating, we have something called the lag phase and the log phase and the stationary phase and the decline phase. So now let's do the characteristics of all. Let's start with the first one, this particular phase where everything is standing still, the lag phase. This is basically all of us in the lockdown. When all of us were in the lockdown, what were we doing? We were sitting, we were eating, we were just sitting at one place, no activity, no moving around. That's it, just sit and eat and become fat. That is what we did in the lockdown. That is what the bacteria are also doing. Although we didn't actually, because most of us were on duties and working. But let's consider it as what most of the people did was sitting and eating and becoming fat at that time. Bacteria in the lag phase, in the lag is just like a lockdown bacteria. It's just going to increase in size. It will gather all its raw materials. It will eat a lot of food. It will make a lot of enzymes. It will make a lot of metabolites. It is going to become very fat. It's going to increase in size. Why is it doing so? I can say it's the silence before the storm. Because after this, it knows I have to work a lot. After this, it knows that I will have to work a lot, multiply a lot. So let me gather all my energy. Let me gather all my energy. Let me adapt and gather enzymes, metabolites so that I'm ready. All my raw material is ready. Then in the next phase, I anyway have to work a lot. So if I ask you a question, that maximum size is at the end of a bacteria. Maximum size of a bacteria is at the end of which phase? We'll know that, okay, at this phase, at this junction, at the end of the lag phase, there is going to be maximum bacterial size that I will encounter. That is the lag. After lag, what do we have next? After lag, the next goes on to this. That is, there's going to be log. There's going to be the log phase. Means exponential. Bacteria has started dividing. There is maximum activity. All the bacteria are dividing. Means all of them are living. All the bacteria are dividing means all of them are living. All of them will take the same color. When all of them are living, all of them will take, like for example, you say, ma'am, I do gram stain. I'll say if all of them are positive, gram positive, all of them will take the color because they are all living. If they are gram negative, all of them will take the color because they are all living. So log basically tells you number increase. There's an increase in the number of the bacteria till a time there comes something known as stationary phase. Can you see? We've entered into a station out here. Why is this a station? Because now some cells have started dying. But we are not going down. If the cells would have been dying, we would have going down. I'm saying we've reached a plateau because right now the live cells and the dead cells are equal in number. That is why we are at a standstill. We are at a plateau because the live cells and the dead cells are equal in number. That is why can I say I have now some living cells, some dead cells. So the kind of staining will be non-uniform. If you remember prior to this, in the log phase, all of them were living. So the staining was very, very uniform. Now I've got some living and some dead, equal in number, which means non-uniform staining will be there. What else? Please remember we write it as stationary phase. We write it as stationary because it's going to tell me that three things happen in this phase. What are the three things? Sporulation. Spore formation happens here. So S for spore formation in stationary phase. Toxin production by the bacteria happens here. T for toxin production. And if, and even bacteria can produce antibiotics. If antibiotic production has to happen, that will also happen in this particular phase. Repeating, guys, if spore formation has to happen, toxin production has to happen, antibiotic production has to happen, all of it happens in the stationary phase, where the live and the dead cells are the equal in number. And finally, you say, ma'am, the cell is dying. When all the cells have died, when all the bacterial cells have died, bacteria have died, when the dead cells are moved, the number will go down. So simple. Now if I get you to the question which I asked you, sporulation occurs in this phase of bacterial growth curve. S for sporulation, what's your answer? Stationary. Because if you remember, sporulation, toxin, antibiotic. Three things happen in the stationary phase. Let's do a last recap. Remember what happens in the lag phase. Lag phase is all about becoming fat. It's all about maximum size. Log phase, finally, the number will increase. Stationary phase, we've already done spore, toxin, antibiotic. Decline phase means everything has basically died. It is all the dead bacteria. That's a bacterial growth curve that they ask you in the exam, which is very, very important. Well, so I can say roughly half of the microbiology is done for us, and in terms of general microbiology. And now in the next session, we'll be taking up the second part of general microbiology, which will include two very important topics. Number one, sterilization and disinfection, and number two, there's going to be bacterial genetics. So two very important ones which I'll be covering up in the second part of the microbiome crash course. Thank you for joining in, friends.